कई बार हो भी जाता, करके देख लो। देख लो करके, 2 घंटे करके दो। हाँ बता दो। हेलो, हेलो, गुड इवनिंग टू ऑल। हाउ आर यू अंकिता? My voice is audible to you? Ankita, my voice is audible? My voice is audible to you, Ankita? All of you, please let me know if you are listening to me. Please let me know. My voice is audible to you. So, Pratishta, please take your headphones along with you. Then you will be able to listen to my voice. Okay, okay. So welcome all of you. Ankita, Kiran, Radhu. Radhu, how are you, Radhu? We have Shivangi along with us, Soumya, Tanvi, all of you. A very good evening to all. How are you all? Everything is fine? And are you able to listen to me? My voice is audible to all of you. Please let me know. Yes, please let me know. My voice is audible to all. Yes, today we have to do the very important topic which you all want to do, which is about this drug discovery, drug designing, all of everything which is related to drugs. Yes, yes. There's an issue regarding the network connectivity. It will be resolved maybe within 5 minutes. We will be able to join the another students also along with us. People is saying, ma'am, what is the last date for submission of the review article? So people basically in my previous lecture series, it is the last day of the session, which is on the 10th day. I told my all students to send me their full review papers. But here I think it's not possible because we are not meeting continuously. There is a gap in between our lecture series. So have you prepared your paper or you are just want to send me your abstract? So if your paper is ready or you are you are doing to make it complete, so you will send me after Diwali. So you have 3-4 days along with you. So after Diwali celebrations, you can send it to me. Okay? Okay, okay, okay. Good, good, good. So we have Priya Shri here. Priyashri, may I allow to ask you what is this RA18110090102221? What is it basically? Hi Ayushi, how are you? Hi Megha. Okay, so Ankita, maybe the issue gets resolved. We will just waiting that it will become okay. Then we will start our today's presentation because today we have to do drug discovery, drug designing. So many things are there related to it. And yes, I forget to send you the PPT of previous lecture because no one of you, messaged me in the morning to send you the PPT and I forget totally about it. So it's my request to all of you when you need something, you can say message me in the morning. Then in the morning, like after this lecture series. I have to take my dinner by 10 to 11. So I'm not free after this lecture series. But in the morning I have not that much busy, so I am able to send you the mail. So please both the PPTs, even the today's presentation and the yesterday's presentation, any of one of you, and then you further send it in a group. Okay, any one of you send it in a group. So tomorrow, any one of you please message me so that I will able to remind it. Okay, so we have Pratik here. So Rupal, have you done your abstract or not? Please let me know. Oh, Ayushi, I'm just saying, just mail me in the morning or just message me in the morning. So Rupal, please let me know, have you selected your topic? Have you done your abstract part or not? Okay, okay, okay. Priya Shrees, that is her registration number. Okay, good, 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 very good. Actually, at this time at 8 p.m., everyone is using, mobiles, and everyone is free and they are using so much of internet that I got this connectivity issue specifically at 8 p.m. There is so much of buffering. All these things causes so much of problem when we are concentrating on a particular subject. It becomes so difficult to concentrate then. But I hope the issue will be resolved by 4 5 minutes maybe. So yes, we have to start with drug designing. So I hope all of you are here, you want to listen, you want to learn something. So shall I share my PPT with all of you? Please let me know if my voice is audible, everything is fine. May I able to share the PPT with all of you? Let me know please. Yes, yes. Okay, now we will start our today's topic. But before the drug part or the drug discovery, drug designing, we have some small. As I already told you yesterday, that we have some special presentation or special PPT on the techniques which are there in proteomics. So these are only few slides. I will cover those slides. Then we will move further to the drugs part. So let me share with you the techniques which are there in the proteomics part. So is it visible to all of you? Please let me know, is it visible? This presentation is visible to you? Gimme, gimme, gimme a first. This is your good name. Gimme, this is your good name. Please let me know. Gimme a first. Is this your good name? And from where you belong to? Please let me know. So this presentation is visible to all of you. Students, please let me know. I hope it is visible. Okay, okay. So now we will start with our techniques which are there in the proteomics session. So I hope you all will be there in the yesterday session also. We have already discussed very briefly about proteomics, the techniques which are there in proteomics, the meaning of proteomics, but I have this presentation also. So I just want to revise, I just want to revise the whole thing which we have done yesterday. So yes, we have proteomics. What is basically proteomics? In which we are dealing with the proteins. So a biotechnology branch concerned with applying the techniques of molecular biology, biochemistry and genetics to analyze the structure, function and interactions of proteins. So when we are specifically working on the protein part, so we have to do the analyzing the structure of the protein, the functioning of the protein and the interaction that is within the protein. So this is important. We have to do the we have to study the structure of the proteins, the functions which they carry, and the interaction of the proteins produced by the genes of a particular cell. So sometimes what happens, we are on a single cell or sometimes we are working on a group of cells. So in the group of cells also or even in the single cell, we have to know the structure of the protein, their functioning and their interaction with each other. So real functional molecules of the cell. So these proteins are the real functional molecules of the cell. Protein-protein interactions of an organism. All these things we will study under proteomics. We already discussed this before also. So these are some terms like proteome, proteomics, protein word, transcriptomics. All these are related to proteins. Okay. But in transcriptomics, often insufficient to study functional aspects of genomics. So all these terms are like genome, transcriptome, proteome, metabolome, they are quite interrelated. Proteomics, organisms have one genome but multiple proteomes. So within single set of genes, we have multiple proteomes. Proteomics is a study of the full complement of proteins at a given time. So this is the important thing, that when we are studying anything about the proteins or proteomes, it is specifically for a time period. Getting my point? So it is a study of a protein at a given time, at a specific time period. So these are a single gene produce multiple proteins. That how from a single gene we are able to get the multiple proteins. Then we have the difference between proteomics and genomics. We already done it yesterday. That proteomics is for the analysis of the protein and genomics is the study of all the genes. So proteomics is specifically for the protein part only and genomics is for the study of all the genes, all the genetic material which is there in the body. So we already discussed it, we will not discuss it today. Yes, these are the three important steps in proteomics. Number one is the purification of the proteins by any method, by any technique. We have to purify the proteins. Then there is a separation of the proteins by gel electrophoresis, by you can say HPLC, by radioactive probes, by any method we have to do the separation. And the last is identification of the protein, in which we have to use mass spectrometry for the identification of a specific protein. Like there are thousands of proteins, but we want to work on a specific protein. So these proteomic tools to study proteins, we have protein isolation, separation and identification. We have to keep it in mind. Then these are the types of proteomics. We have expression proteomics, we have structural proteomics, in which we are studying the structure of the protein. Interaction proteomics, when there is an interaction between different proteins, or you can say it is a protein-protein interactions basically. These are the techniques which are used in proteomics. We have mass spectroscopy, we have microarray technology, we have bioinformatics, we have blotting. All these things are used for the proteomics study. I will share the PPT. You will just go through all these techniques, 2D electrophoresis, what is gel, like we have agarose gel, we have polyacrylamide gel, we have different types of gels and they have some different functionings also. So there is a difference between agarose gel and polyacrylamide gel. So you have to know the difference between both of these two. Okay. So this is 2D gel electrophoresis, and we are able to get number of proteins. The pink one, the blue one, yellow one, white one. They are representing the proteins according to their molecular weight. So they are representing the proteins. Then this is about the gel electrophoresis basically. So you just go with all these techniques and let me know if you are not able to get anything. Then microarray is there, which we have done yesterday also. Like we have DNA microarrays and we also have the protein microarrays. After it we have mass spectrometry. Mass spectrometry is very important in proteomics. It is very important in proteomics. It is an instrumental technique to rapidly moving positive ions by electron bombardment, and charged particles are separated according to their masses. So the important point is the separation is based on the mass to charge ratio. So there is a basically the separation is based on the mass to charge ratio. So we have to know the ratio of mass to charge. So they have some principle that how the ions will migrate differently or how they will attract toward the positive side or the negative side. So this is mass spectrometry. I think any one of you must have done this also. We have HPLC, which is high pressure liquid chromatography for the separation of the proteins. I will not discuss it today because we already discussed it yesterday. Then we have master layout, which is again a technique for the separation of the peptides. You can say, if you are able to see, there is a mixed sample. Then after the purification, we are able to get the peptides differently. These are the based on the chromatographical technique. So I will share this PT. Just go with it. If you are unable to get anything, please let me know. We will discuss definitely. So that is all in this technique. I hope you are able to get it. Okay, I will share this proteomics part, the techniques which are there in proteomics. It is only left yesterday, so I think let me show you only one. I will share with all of you and you have to read it thoroughly. If I share to you, if there is any doubt, then you have to let me know. Getting my point? So now we have to move to our main topic, which is basically the drug discovery. So we have to start with drug discovery, drug designing. All these things are very important and definitely they are correlated. So I will try my best to tell you about this drug discovery, drug designing and all. So are you able to get it? Please let me know. Any one of you, please let me know. Yes, my voice is audible. Please, anyone, please reply to me. Okay, okay. Pratik is saying yes, ma'am. Okay, thank you, Pratik. So let's start further our main topic, which is drug designing and drug development, drug discovery also. Let me share the PPT with all of you. Yes. So I have two presentations on these drugs, drug designing, drug discovery. I will try to do both these presentations today. So let's start. What is drug discovery and what is the developmental process of doing this? So some topics are there which we have to cover. The introduction to drug discovery, procedure for drug discovery, drug development, the steps which are involved in drug development, preclinical evaluation and clinical evaluation. So this is the drug discovery cycle. You are all are able to know what is drugs basically, the medicines which we are using, the chemicals which we are using, how we are able to discover those chemicals and how we are able to implement those chemicals in our daily nature or in our daily activities. So this is the life cycle, how there is a designing of the drugs, how there is a synthesis of the drugs, and which are the parameters which are there for the evaluation of the drug. So first of all, let me tell you what is the drug and why why we are using all these things. So in the ancient times, most of the drugs used in the treatment of diseases were derived from naturally occurring substances of plant origin. So in the very ancient times, what is happening? We were using the plant origin chemicals like we are getting opine from poppy, quinine from cinchona, and similarly digitalis from foxglove. So we are getting the chemicals from the plant sources. But now, presently, the majority of these practic agents are synthetic in nature. So nowadays we are using the medicines or you can say drugs which are chemical in nature. But in the ancient times we are using the chemicals which are taken from or which are you can say derived from the plant. So drug discovery and development is complex. So we have to discuss about drug discovery and their processing, their development. It is a complex, time-consuming, costly process which carries commercial risk. Suppose you have made some drugs, you have made some corona vaccines for example, something like that, but it also carries some commercial risk. Okay, how the public will be able to use those chemicals, what are the limitations of those chemicals, so everything has to be taken in mind. So drug discovery and development is broadly divided into three main components, which is drug discovery. What happened to the presentation? So in the drug discovery, what happens? Of course, when there is a disease, suppose there is a pandemic or some type of, you can say the disease is there, then we have to search for a new drug. Okay, so this is drug discovery, that many tests of molecular compounds to find possible beneficial effects against any of large number of diseases. When the disease becomes pandemic, so much large in number, so we have to search for a new drug. Existing treatments that have been unanticipated effects, new technologies such as those that provide new ways to target medical products to specific sites within the body or to manipulate the genetic material. So we have drug discovery, or you can see the need of doing this drug discovery. So, development once researchers identify a promising compound for development, they conduct experiments to gather information on how it is absorbed, distributed, metabolized and excreted in the body. So we have to keep all these things, that how the chemical will be absorbed and how it will act on the target site, how it will metabolize further and how it will be excreted out of the body, its potential benefits and mechanisms of action, the best dosage, the best way to give the drug. So we have to think about all these parameters, that the benefits of those, chemicals or compounds, the best dosage, that how much quantity we have to give, the best way to give the drug, we have to keep all these things in our mind. Side effects or adverse effects that can often be referred as toxicity. Yes, this is very important term. Toxicity. What do you mean by toxicity? Toxicity is basically any type of compound or a chemical which when we will take in a limit it's okay, but when we will take it out of the limit or excess of the limit, it leads to the death of the person. So this is toxicity. How it affects different groups of people and such as gender, race or ethnicity, how it interacts with other drugs and treatments, its effectiveness as compared with similar drugs. So all these things, all these parameters we have to keep in mind when we are going with the drugs. When we want to drug discovery is different thing, but when we want to do the development, so we have to go through all these parameters. Okay. So these parameters you have to keep in mind when we are doing any type of drug discovery or something. We have to keep all these things in mind. So this is the procedure for drug discovery, that how we are able to do the drug discovery. Like first is random, that you are able to get any chemical or compound, or you are able to know the functioning of any chemical. Then another is molecular manipulation, molecular designing, metabolites of drugs, serendipity. All these are the parameters or the procedures for the drug discovery. Okay. Then what is random screening? What is random screening? So IITs are subjected to the battery of screening tests designed to determine different types of biological activity. Such tests include studies on animal behavior. Yes, this is very important. This is very important that we have to know that, you know, if we are injecting that particular chemical or compound, how this chemical or compound will affect the biological activity of that particular organism. So such tests include studies on the animal behavior, isolated tissues, intact animal, and sometimes even an animal model of a disease. Such studies are time consuming, expensive, and have a low yield. So when we are doing random screening, what will happen? When we want to introduce some chemical, we want to introduce some compound or something like that. First of all, we have to properly check the biological effect on the particular organism. Suppose we are injecting that particular compound or particular chemical in the mice or in the rat for experiment purposes only. Then after injecting that particular, you can say, compound or a chemical, we have to check the behavior of that animal. That means of the of the mice or of the rat, we have to check their behavior, the changes which are there in their behavior. So this is random screening. Then we have molecular manipulation also. This is more logical approach and may yield new compounds. I don't know why this presentation becomes so hotspot. I don't know. So this is metabolites of drug. This is serendipity. Or serendipity, what do you mean by? It means happy observation by chance and has led to introduction of many remedies in the past. Example, use of organomolecules for cardiac edema, penicillin as an antibacterial agent. So what are basically serendipity? It is a happy observation by chance, which happens by chance. So drug development, once a new chemical entity is discovered, it has to be subjected to the development. When we discover some chemical or a compound, it has to be go further for the processing which is there. There are some parameters which we have to think of. Like these are the some steps which are there in the drug development. So first of all is we are able to do the drug discovery, then there is another term which is drug development. Like here there are some parameters, some points are there by which we have to follow, like preclinical synthesis and physiochemical analysis, then preliminary biological analysis, or you can say preliminary biological evaluation, then secondary and specific biological evaluation. We have to also study their toxicological studies. So all these parameters we have to do. This is phase 1, then we have to move further to phase 2, then we have phase 3. Lots of so many parameters are there when you are doing the drug designing, or you can say drug development. So lots of phases are there. Like drug discovery research, we have screening of these things, preclinical research, clinical development, all these things are there for the drug development. Like the last step is post marketing drug development, but before it we have you can say production and quality assurance, all these parameters are there, then we are able to get the final product. Okay. Yes, this is the definition of clinical trial, like clinical pharmacology. What is clinical trial? A careful and ethnically designed human experiment with the aim of answering some precisely framed questions. So, what is basically clinical trial? When we are doing something, when we are using humans as experiment, but ethnically, if you are, even if you have to use mice and rats, you have to take a specific permission from the specific committee. There are some rules. Okay, when you are using any type of drug to any organism or anyone, there are some rules which we have to follow. So there are some ethics and we have to, the patient selection is also there. Experimental design. The design of the trial should be preferably a biostatic, should be consulted. In general, controlled clinical trials must include 4 safeguards against the bias. So we have some, you can say some parameters, like we have double blind technique, we have randomization of treatment, all these are the phases of the clinical trials. We will not discuss it. So this is again drug discovery. So drug is basically what? Any chemical substance of known structure other than a nutrient or essential dietary ingredient, which when administered to the living organism produce a biological effect. So when you are introducing the drug into the living organism, it will definitely produce some effect in your body, then it is known as the drug. Other than the nutrients and basic dietary ingredient, which when administered to the living organism produces a biological effect. Are you able to get a biological effect? That means it has some effect on our body. So identification of new chemical entities as a potential therapeutic agent. This is the discovery phase when we are able to get the chemical, we are able to found the compound. This is the discovery phase, and then another is the development phase when the compound is tested for the safety and efficacy for one or more clinical indications. So this is known as the discovery phase and development phase. So the term drug discovery is classical and conventional. It should be replaced with the term drug invention. So drug discovery is basically drug invention, when we are able to found a certain chemical or compound which is used as a drug. This is the thing, that drugs were discovered from the natural resources in the previous times, but now synthesizing, or we are synthesizing chemically also. Now we have new drug definition which is given by the Government of India according to the rules 1 to 2E. A new substance of chemical, biological or biotechnological origin in bulk or prepared dosage form used for prevention, diagnosis, or treatment of diseases in man or animals, which, except during local clinical trials, has not been used in the country to any significant extent. That means the drug is not previously used that much. So this is the new definition of the drugs by the government of India. So a drug already approved by the licensing authority, which is now proposed to be marketed with the modified or new indications, dosage form, and route of administration. So these are the definitions of drug. Kindly read it by your own self. So we have drug discovery, we have preclinical development, in which we are doing the toxicity test, pharmacokinetic analysis, and formulation. Then we have clinical development. The selected compound is tested for efficacy, side effects, and potential dangers in the volunteers, or you can say the patient. So there are some stages of all these drug synthesis or drugs when it is introduced into the biological form. So we have drug discovery when we are able to form the compound. We have preclinical development. We have some phases. First is preclinical development, to check all these things like toxicology. Then we have clinical development, which has three phases. Then we have regulatory approval, and the last stage is the post-marketing surveillance. So all these things are there. We approximately need 2 to 5 years. Getting my point? 2 to 5 years, or it may take also more times also, like for the COVID vaccine, for this coronavirus, we are not able to find the drug yet. So it definitely takes 2 to 5 years. Then we have preclinical development, which approximately takes 1.5 years. And then later, the clinical phase is too long, for 5 to 7 years. We have to do lots of experiments on the clinical basis. Then it is approved by 1 or 2 years. And at the end, like suppose we have 100 compounds, and at the end we are able to get the one compound or one drug specifically for the disease or maybe for something. So only from 100 projects we are able to get only one drug which pass all the parameters. So let me check whether you are here or not. Are you able to get my point? That yes, I will share the PPT. No issues at all, dear. I will share it. Don't say this thing. I will share it. So you understand this, the parameter of this diagram, that how in how many stages we have to cover for final approving any drug or any compound or any stages. After it, like we have 100 compounds or chemicals here, but at the end we are able to get only one compound, or you can say one drug basically. So are you able to get it? PPT is not clear, but why it is not clear? It should be clear, everything is clear. It is not clear now. Please let me know. Please let me know if it's clear to you or not. PPT is clear to you or not? Are you able to listen to me? Please reply if you are able to listen to me. Please reply if you are able to listen to me. Any one of you? So are you able to listen to me? Please reply. We will definitely you can say we will shift it to Zoom or Google Meet from tomorrow. No issues at all. If there is so much problem regarding the presentation or even it is not visible, then how we will do it further. So it's no issues. We will do it on Google Meet or Zoom. No issues at all. But for the now for today's session, please let me know. Are you able to understand this image which I showed to all of you? Is it clear to you? Please let me know. Any one of you. So shall I continue now? Please, if you are able to listen to me, please let me know. My voice is audible. Shall I continue? Please let me know. Audible is okay, but are you able to listen to? Are you able to see the presentation also? Please let me know. Okay. Okay. Otherwise, if there is problem regarding it, we will conduct it on Google Meet from tomorrow, not today, because now we are doing the presentation right now. So from tomorrow onwards, we will take it on Google Meet or on Zoom also. No issues, okay? Don't worry, we will do it on that platform also. So yes, we are discussing about these things, which is drug discovery. As we already told you that approximately we have 100 projects when we are doing the drug discovery. We have 100 projects, or you can say 100 chemicals or 100 compounds. But from all those compounds, we are able to get the only one or two drugs at the final stage. So at the final one or two products which is approved for marketing, which is approved for you can say passing to the public, only one or two are approved. So what is basically the drug discovery? Yeah, the time duration for drug discovery is basically 2 to 5 years. So it approximately takes 2 to 5 years when we want to discover something, when we want to make a drug for some disease or something. It will definitely takes 2 to 5 years, and the number of projects or number of compound is 100. It consists of following components, which is target selection and validation, lead finding or lead generation, lead optimization, pharmacological profiling. So these all are on which you can say on which target the drug will act. All these things have to keep in mind. We will discuss all these things briefly also. Then we have one more term. It is a hypothesis which is first made that modulation of the activity of a specific protein target will have beneficial therapeutic effects. The reverse pharmacology approach takes about 2 years to obtain a new drug candidate. It is much faster and more efficient. So we have two types of approaches. We have classical pharmacology. So in classical approach, this is the general part or the general approach which is there in pharmaceutical sciences. Pharmaceutical, he or she will definitely know about it. That we have these things which we keep in mind or something, like first of all functional activity on which the ligand will act, and then biological role or pathophysiology. All these things are there in the pharmacology. But when we are using what we are using bioinformatics. We will discuss in detail also. So when we are using reverse pharmacology, we are using the bioinformatics as a source also. So this is the difference which is there in the regular pharmacology or regular drug designing and the reverse pharmacology. So, properties of ideal drug target. So the target is a disease modifying or has a proven function in the pathophysiology of a disease. The modulation of the target is less important under physiological conditions. The target has a favorable accessibility enabling high throughput screening. I just want to show you reverse pharmacology in detail. Just let me check it. So these are the targets of drugs that on which area these drugs will act. Like they will work on the G protein coupled receptor or on the nuclear receptors or on the ion channels or on the enzymes. That means on which target these drugs will work. Okay, so we have receptors which are present in our body. That means on which receptors these targets or these drugs will work. Okay, so we are exploring only 120 distinct drug targets. So till now we are only able to know about 120 types of receptors or channels on which these drugs work. One minute, just let me check. Here biotechnology is also helpful. We have immunopharmacology. I just want to show you something. We are using traditional medicines, when we are using traditional any chemical, like we are using papaver somniferum for morphine, using till now, like we are using aspirin. So all these are from the plant sources, like this is from willow bark. We have quinine which we takes from the cinchona tree. We take this drug from the plant which is Atropa belladonna. So from Atropa belladonna we are able to get the drug which is atropine. Similarly aspirin is here, morphine is here. So these are the plant sources from where we are able to get the drugs or compounds or you can say chemicals. So these are the animal sources from where, like this is alpha bungranotoxin, alpha bungrotoxin, which we are able to get from the snakes. So from snake's poison we are able to get this chemical or compound you can say. Similarly from rabinos, from dinoflagellates which are there in the water, which are there in the oceans, we are able to get the saxitoxin. So these are the chemical compounds which we are able to get from the animals. So these are the sources like animals and plants. From these animals and plants we